Well, thanks everybody. I'm glad to be back. I'm going to talk to you about something a little different today than I did last time, which is about one of our really special local salamanders, which is called the Santa Cruz Long-toed Salamander. And I see a lot of familiar faces, but in case you don't know about me, as Dylan said, my name's Regina, and I'm doing my PhD at UC Santa Cruz. I'm almost done now. When I last talked to you, I just started. And my last talk was all about my general research, which is more about how amphibians in general, across different kinds of species, are affected by climate change and their potential adaptations to it. So if they can acclimate or migrate, things like that. And I'm still doing that work. But on top of that, I've started this new project in collaboration with US Fish and Wildlife about actually conserving one of the local species of the same family of salamanders I work with, using the methods that I've been using in my PhD. And so that's what I'm going to focus on today. But before I actually give you information about the salamander, I want to just kind of remind everybody about the plight they're facing. So I won't give you the whole spiel about how climate change is bad and that hurts amphibians. I hope we all know that at this point. But one of the misconceptions about especially ectotherms, the things that are thought of as cold-blooded, like reptiles and amphibians, a lot of people think the danger of climate change is it will get so hot that they'll just pass out and die. That is possible, but what is more likely instead is that as temperatures rise and other things happen with climate change, the conditions just won't be as ideal and populations will start to shrink. More animals die off or can't mate or can't find breeding partners. And what happens is the population shrinks into extinction. And so that's what a lot of our local amphibians are facing. So I'm going to talk about what we call an extinction vortex, which is what this species, the Santa Cruz Long-toed Salamander, is facing. So an extinction vortex is this kind of cyclic event we're going to talk about. Populations start having shrinked, like being shrunk for some reason. For our species, it's habitat loss. For others, it could be a disease or just general climate change practices, but populations shrink. When a population is small, something occurs that we call losing genetic diversity. So what that means is all populations of animals have genes, right, genetics, and the entire genome across all species, all individuals in a population we call the genetic diversity. How many types of genes they have, the different versions of genes, things like that. When a population is really small, they're at risk of losing genetic diversity. First off, there's fewer individuals to breed to get genetic diversity, but they're also at risk of things like disasters shrinking genetic diversity, or there's lack of what we call gene flow. They can't find or mate with somebody else. And what that means is they're losing more and more genetic diversity. The reason that is bad, there we go, is populations with very low genetic diversity aren't as able to adapt or recover from disasters or changes in their environment. They cannot recover from things like habitat loss or climate change. They've lost the diversity needed to adapt to these problems. What that means is they can't recover from things like climate change. Their risk of extinction goes up. They are more likely to risk of extinction is what it sounds like. They're more likely to go extinct and their population will shrink. And that creates what we call the cyclic event or extinction vortex. It works like a hurricane, excuse me, where the population is shrinking. It loses genetic diversity. It can no longer recover or adapt. So it goes more and more extinct. It shrinks, and this cycles and cycles and cycles into a vortex. And that's really hard to break out of. So the species we're going to talk about today, the Santa Cruz Long-toed Salamander, is in what we call an extinction vortex. They're unable to escape this currently. Today, I'm going to talk about how we know that and what our plans are to fix it. I am going to talk a little bit just about the Santa Cruz Long-toed Salamander. It's a really important species, but very little is known about it, both to scientists and also to the general public, for many reasons. So part of today is just going to be me explaining to you a little bit about the long-toed salamander and also about seeing what we're learning about them. So this is this little guy over here. This is what an adult looks like. It's very pretty. Has this kind of like, I don't know, from head to like back feet, it's about 4 inches long maybe, and then another 4 inches of tail. So they're a little small. Their Latin name is Ambystoma macrodactylum crucium. So technically they're a subspecies, although they're treated like a species for my talk today, and also how our government kind of treats them. Their spots are all on their back, and they're kind of their like key factor is this bright yellow spots. Sometimes they're even more yellow. They're kind of gold-like here on their backside. So this picture is actually like from top to, it's a bird's eye view photo of the salamander. The Santa Cruz Long-toed Salamander is something that we call highly endemic. And what that means is their range is very, very small, and they have a very, very focused area that they live. And what that is for us is they are only native to Santa Cruz and Monterey counties, and not just the whole counties, just the south half of Santa Cruz and the north half of Monterey. They have one of the smallest ranges of any vertebrates. And they are also, in our case, endangered. They have full endangered status by the federal government. They're actually one of the first animals on the endangered species list. They were on the list before it was even called the endangered species list, when it was called I believe it was the Endangered Conservation Act. So they were put on in the 60s. And on top of all of that, they have an even extra special status called fully protected, which is not a status of endangerment that's even given to species anymore. It means they have the highest protection you can possibly give a species. These guys have at both our state and federal level. The question in the chat that I think is a good time to answer here is, how does they become so localized or endemic? In this case, the nearest sister subspecies to this guy is the southern long-toed salamander, which is in like the Sierras, kind of the mid-Sierras or South Sierras. And the reason that they have separated evolutionarily is the formation of the valley in between us. So at some point that valley formed and separated our two species. And eventually they evolved to be separate subspecies. So that's why their range is only localized to here and not other places. All right, let's talk a little bit more about these guys. They have, just to kind of remind you if you're unfamiliar with the life history of the salamander, because it's going to be important to what we know. These are all pictures of tiger salamanders, because I have some of the first pictures of eggs, which is very exciting, that I'll show you later. But they follow what we call the life cycle of a terrestrial species. So they have both a terrestrial on land phase and an aquatic phase. So the way this works is adults live in kind of like forested, what we call upland areas, and they migrate down to breeding ponds during the rainy seasons. So now, adults, males and females, come to the ponds. They mate in the ponds and they lay aquatic eggs. So these eggs in this photo here are actually underwater. The coolest thing I think about this family is their eggs, and most amphibian eggs, to be honest. Their eggs are actually see-through, and you can see the larva developing in them. So these kind of C shapes are the larva going through the different developmental stages. So then what happens later, kind of late winter, early spring, is the eggs hatch into these little aquatic larvae. They have these gills that help them breathe water. They're eating like invertebrates in the pond, and they live in there until early or late summer, depending on the location. And they then do something we call metamorphosis. And they go through this transition phase where they lose their gills, they actually grow lungs, and they become terrestrial adults. And once they leave the pond, they migrate into the forested upland habitat, and the cycle kind of continues. And I want you to think about this as we go through, because when we talk about conserving a species, we have to think about conserving each life stage. All right. I also just kind of want you to think about what the ponds look like. So when I think of a pond, I think of some beautiful body of water. That's not what most of these look like. This is a pond. If you can spot it, you get extra points. But right here is land. And where all these kind of like reed-like plants are is the pond itself. So adults are coming, they can travel sometimes, we think, we don't know a ton, but over a mile to get to breeding ponds. And adults, we think, as you'll learn, we don't know a lot about the species. And that's part of what I'm going to talk about today is the things we found out. But the adults come to the pond where they were born to breed. And they breed in this kind of marshy habitat, which is really good. It has cover for babies, all kinds of plants to grow eggs on. But this is what we're talking about when we're thinking of a pond here locally in our counties. So I want to talk about a little bit the history of the long-toed salamander. It's going to help you set up why they're so endangered. So this is just a map right here. To rearrange you, Santa Cruz is here, Monterey is down here. We've got Watsonville smack in the middle. This orange is their entire population range. Might feel big to us, but that's very small for a species here. I believe it's like 15 miles by 5. It's like, I don't remember exactly how large it is, but it's small, very small. And you can kind of see the county line somewhere around here, I believe. And when humans didn't live here, that was a lot of nice connected habitats. Since they do have to migrate to ponds, they have to be able to get to the ponds. But over time, we have a lot of housing development in this area. There's a lot of agricultural development. And, you know, really important, this Highway 1 that goes right in the middle of their range has caused fragmented populations. So salamanders are small. They can't cross Highway 1. They can't cross major sloughs. They can barely kind of cross through their normal habitat. So populations that were once well connected are now separated. When a population like this is separated into really distinct kind of like subpopulations where they can no longer kind of get to each other, we call them metapopulations. So we now have, I believe it's 6 distinct metapopulations where they're no longer sharing genetics. And this has caused this kind of major habitat loss is the major factor for why they have become an endangered species. So over the last 50 years is actually when a lot of this has happened. Or at least the more recent separations. So this is the same map, kind of the same scale too, where we have Santa Cruz up here, Monterey down here. But what these black dots show you are active breeding ponds. So they're breeding ponds where we still see a lot of breeding. And as you can see, there are some populations up here in like Aptos. So if you're by like Seascape, Rio Del Mar area, they're separated from the freeway from who they used to be able to get to up here in like the Calabasas-Larkins Valley area. And then populations here are separated from the freeway, and they're also separated up here by housing developments. And then we also have the slough. And this slough and populations on either side of the sloughs are also going to be stuck in the sense that they can't migrate to each other. And as I mentioned earlier, what this means is they're all becoming genetically distinct. They're becoming genetically cut off from each other. And some of these populations, especially the ones down here in the south, in Monterey, are losing genetic diversity. So another graduate student, many, many moons ago, did do a genetic study to confirm what I'm saying is true, that these kind of sets of populations are all genetically separated. They can no longer, there's no what we call gene flow. There's no genetics coming across populations. And that these ones in the south, not only are they genetically separate, they are also losing genetic diversity. So a reminder that is bad when it comes to adapting against climate change and can lead to extinction. And what is happening is these ponds specifically, they're all shrinking, but very, very specifically, the Monterey County ponds are really shrinking. There's becoming less and less adults at each pond every year, and enough that we're worried. We're very worried. And fish and wildlife, I don't actually know if what I'm going to say is true, but in my mind, in my experience, this is one of the most heavily monitored species I've ever really seen in California. I think part of it's because we can actually measure the whole range, and Fish and Wildlife can actually track or try and track every adult in the entire species. there are so many conservation methods going on. There are so many biologists for U.S. Fish and Wildlife, California Department of Fish and Wildlife. Also, there's a lot of private biologists who are hired by different like consulting firms, things like that, who are doing things to try and make this better, to make these populations, one, more connective, so they could actually maybe breed across and share genetics, and also just to increase population numbers. So I have a picture here of one of the things they're doing. I've seen this the most in kind of the Aptos area, is they're literally building tunnels under roads and bridges over roads. So for example, I believe, I can't remember if this is the exact one, but there's like 2 ponds in Aptos that are not that far apart, like maybe a quarter of a mile, maybe even less than that. But they're separated by a giant road and 10 houses. And for a baby salamander, well, not a baby salamander, adult, but tiny salamander, they can't cross that. So what they've been doing is building these tunnels and these kind of funnels that force animals to go under the tunnel to kind of make some of these populations connective. They're also doing all kinds of work removing removing invasive plants and planting natural plants so the habitat is correct. They're also monitoring population size. They cycle every year and try and capture the adults at every adult at each breeding pond every few years, so we can kind of have an estimate on breeding size. It sounds like that'd be easy to do, but it's hard since these adults live in the forest most of the time. We can't really find them. So the easiest time to track them is when they come to breed, and they all come and collect at different ponds. Fish and Wildlife is also building new ponds, places where they has great habitat, and maybe a pond would help kind of connect 2 farther apart ponds. They're building ponds. They're also restructuring current ponds so their water tables last longer into the year, so larvae can hatch. They're buying all kinds of private land so that they can preserve different pathways for these salamanders. And this is pretty intense and awesome, I think. But part of the problem is, even with all of this, it hasn't been enough for those southern populations. We still see them shrinking and shrinking, and we know it's because of a loss of genetic diversity. So what we decided to do, and the project that I'm going to talk to you about today, is we decided to breed across these populations and actually kind of force what we would call gene flow, so to increase genetic diversity. And that's the main project I'm going to talk to you about today. So our goal, besides just releasing more larvae into the wild to boost population size, was also to boost genetic diversity. And so the way that we do that is by breeding across different populations. So what we did is we collected adults from the healthy northern populations and adults from the southern struggling populations and bred them together, literally mashing their genes together to increase genetic diversity of their larvae. That's all great. We could release a bunch of larvae into struggling populations. We could reduce a bunch of genetically diverse larvae into a bunch of struggling populations. But there's a lot of also concerns about that. You don't want to just release animals all willy-nilly, mix up the genetics. Most of the time, natural selection has had a reason to form genetics the way they are. So what we had to do before we could do all this is test that this was a good idea and make sure that the larvae we were going to breed were what we call fit, that they would be able to survive and reproduce in the wild. The thing we are specifically looking for is what we call outbreeding depression. So you may have heard or have heard something about inbreeding. So, you know, we all know you're not supposed to marry your cousin, right? Inbreeding is bad. It has all kinds of negative impacts on health. It also decreases genetic diversity. But we also have the opposite, which is what we call outbreeding depression. If populations have been separated for long enough, even if they're the same species and can breed, sometimes there can also be negative effects, like weird mutations or adaptations that are counter each other out. So one of the things that we had to do before we just bred across populations and release is check if that was a good idea by looking and making sure that there was no what we call outbreeding depression. And if that was true, then we'd be able to actually release the larvae in ponds that are new ponds that are both being built and ponds that are struggling. So I'm going to talk a little bit about the methods, and the first one being how we get the adults. So what happens when we need adults is Fish and Wildlife puts up these huge, what we call drift fences. What's in this right picture here. So this fence would actually be, this is not one of their fences. I stole this picture off the internet. As you can see, it looks kind of tropical, but this fence comes around the entire pond. It circumferences the whole pond. And so what happens is when adults are coming to migrate to the pond, since we don't really know where they come from, they hit this fence, and what they do is they move along the fence. And as they're moving along the fence trying to find a way in, they fall into a bucket. And you can see this is a photo of when they're catching frogs. This is a very safe method to capture adults, and in theory, we would catch every single adult that moves into a breeding pond, and we can both count the numbers of females and males, and also, in our case, collect adults for our project. So what we did is what we call an admixture breeding design. So we couldn't just breed these crosses that we're talking about and test for outbreeding depression, because you need something to compare them to. So what we did is we bred the healthy animals from Santa Cruz. We bred pure strains of them. We bred pure strains of the struggling Monterey populations. And then we also bred pairs of animals that were crosses, both female Santa Cruz animals to male Monterey animals, and also male Monterey animals to female Santa Cruz. And what that gives us is this admixture design. So we can test, is there impacts on breeding compared to the pure strained animals? So we call that like a pure strain. We'll go through this. So as I said earlier, the adults migrate in winter. So about now is when adults are getting to ponds. It's toward the end, and laying their eggs about now. So we've done this for 2 years so far. And so adults were brought in. Well, before adults were brought in, let's talk about the breeding tanks. Step ahead of myself. So these are what the breeding tanks look like. They're not very pretty, but they're very functional. They're these large, I believe they're 300-gallon tanks. And I think their original purpose was like cattle troughs, but they work for us. And so what we did before we even brought adults in is we did something we call seeding. So one of my personal concerns doing this, I should mention, right, this has never ever been done before, so we were trying to play it safe, was we needed natural conditions for the animals. Salamanders are, as amphibians, are sensitive, right, to different water conditions. And little itty bitty larvae, once they hatch, are probably sensitive to what they eat. And those are not things that we can just like buy at Petco. So what we did is something we call seeding, which is in bringing most of the environment from their habitat here. So what this tank looks like, it's a little hard to see with the reflection, maybe you can see better over here, is we actually dug up plants from the natural ponds, plopped one in every single tank, filled the bottom with mud from the natural ponds, and sticks and rocks and all kinds of like that, the gross gunk. And we let it sit for a long time. And what that does is creates its own little ecosystem. So what we have in here not only is mud and plants and rocks from the environment, but when you bring the mud, you bring all the little bugs and critters and algae and all that with us. And what we did was create our little own ecosystem. So we had all kinds of what we call invertebrates in there, all kinds of algae and plant growth, so that the water conditions would be held steady. When it cycles like that, the water actually stays nice and clear and nice and low in like the sulfates and sulfites, the things you try and worry about when you have a fish tank. And so we did that first. What we did then was bring in adults, as I told you, from this, some from Santa Cruz County, the healthy populations, and some from Monterey County, the struggling populations. And we made pairs of breeding right here. I think, let's see, I think I have more on my next slide. I can move it. No, we'll keep talking about this side. And so adults were placed in these tanks. They move as soon as the like second rains hit. So somewhere in December or January, adults are collected, put in here. Some of this was all a little trial and error, so we learned as we went. Females in this species have what we call female choice. They won't mate with just any male. They have to have a nice, prim and proper, pretty male. So females were put with multiple males, so they would have a choice. When they chose the male to mate with, they would lay their eggs. So once mating occurs, eggs are actually laid in about a day or two. The females already have the eggs ready to go. The females lay all of their eggs, and then the adults are released back into the wild. So we did not keep the adults for very long, just as long as it took for them to settle in and breed. And they all got released where they came from. Then the eggs stick around, depending on the temperature, from about 3 to 6 weeks, and then they hatch into these little larvae. They're really small. I'm going to show you some pictures later. And then we kept the larvae until they are almost ready to go through metamorphosis. So that's that stage where they become an adult. So we kept these larvae in these tanks growing and feeding them and getting them nice, big, and happy, and then put them back in their ponds before they metamorphosed. So next I'm going to talk about the tests that we actually ran to see if this was a good idea, to see if there was any outbreeding depression. So the first thing we did is actually test what we call the fitness of the offspring. So that's both, these are all kind of measures of survival fitness. So how well can they survive in their habitat? So we measured things like the rate of egg fertilization. So is there a difference between the fertilization rate of like a pure healthy population or the non-healthy population, or the crosses between the two? You can see here that these are eggs. These are eggs laid the same day. These all are. So you can see this outside here is a protective jelly. It keeps the egg safe. The actual like shell of the egg is this inner clear layer here. And then this whitish brown thing is the actual embryo itself. So we measured the number of eggs and how many eggs were fertilized. Then we also measured the successful hatching rate of the eggs. So how many eggs were able to make it from fertilization to hatching. And then we also measured some things about the larvae. So these are larvae that are probably about a month old at this point. You can see they don't really have any legs yet. They're mostly just heads and tails. The legs come later. Some of them have just started developing arms. I'll show you a closer up photo later. But the other thing that we measured to see if this cross-testing was effective was the growth rate of the animals. So we measured the animals every week to see if there's a difference in growth rate, and also survival rates of all of the larvae. And then what we also did was just test the larval diet. So as I mentioned, not very much is known about the species. And this is the first time they've ever really been brought into captivity. So we were also just trying to learn as much as we possibly could about what we call the natural history of these animals, just their general life patterns. And since we brought all of the mud and sticks to these tanks, all of the invertebrates in the tanks were natural to their environment. So what we also tested was, what are they eating? Do they have certain organisms they prefer to eat? Will they eat anything? And also how fast they can eat, which is very fast. So what we did every week was measure changes in what we call the invertebrate species in the ponds. So these are just one species right here. These little bulbous things are Daphnia. They're microscopic. This is under a microscope. So we can't really see them, but the larvae can see them and eat them. We measured all kinds of different invertebrates and also over time. So we took measurements of the invertebrate colonies every week and measured how fast and how much they were eating, but also what they ate as they got larger. So as things started to disappear, we made the assumption they were eating them. We can see tracks of what species they prefer to eat when they're small versus medium size versus large size. Some other tests that we ran, and this is where my research kind of came in and why we were asked to do this project, is measuring the fitness of their physiology. So their actual like ability to adapt to thermal changes. One of the measurements that we took is something we call thermal preference. That is the temperature an animal wants to be at. Like they would prefer to thermoregulate at that temperature, giving a large range of temperatures. This is kind of what our setup looks like. I don't have a picture of the real setup, but we create this long aquatic track that starts at 30 degrees Celsius. That's pretty hot. That's like 90, 95, down to 10 degrees Celsius, which is really cool. And we allow the animal, this is a picture of a little larva, to thermoregulate just naturally this environment, and we track which temperatures they want to live at. That helps us know if the larva would be able to handle being released into certain ponds. All the ponds around here have different temperatures that they get to each year. Some get higher than others, and this kind of allows us to see where the potential release sites could be. Another measure of thermal physiology that we measured is something we call the thermal performance. So all animals, us as humans included, have what we call a thermal performance curve. This is just a cartoon of one, where we have body temperatures, and at different levels of that temperature, we have different performance rates, right? So if we want to talk about, let's talk about humans running. When our body temperature is really, really cold, we can't run very well. There is some optimum temperature where our body would be at that we could run very, very well, our peak performance. And as our body temperature got hotter, once again, our performance would kind of decrease. For us as mammals, and what we would call an endotherm, so like a warm-blooded creature, we regulate our own body temperature. But for amphibians and other ectotherms, or what we call cold-blooded creatures, they can't. So they have to use the environment to adjust their body temperatures. So if you throw one of these little larvae in a 5-degree bin of water, that's what its body temperature is going to be at, and its performance might be low. So what we did is we actually created this performance curve for each of the larvae. And our measure of performance for the larva is how fast they can swim. The biggest thing you have to worry about when you're a larva is not getting eaten by birds, other reptiles, and even more importantly, by your siblings. This is a very cannibalistic species. So what we tried to do is measure how fast they could run at a range of at a range of degrees. So we measured how fast they could swim at 5 degrees, 10, 15, 20, and 25, I believe. And what we can do is create this curve. I want to show you, because people always ask, how do you make a larva swim? The answer is pretty funny. Let's see if the video will pop up. Yes. So for reference, this is just a tank with a grid on the bottom so we can measure distance. And this is the little larva. We did this with the larger larva so they're nice and sturdy. You can maybe see there's legs now. And what we do is we keep them at these temperatures and then see how fast they can swim. And we do that by poking them with a little paintbrush. So let's see if the video will play. There we go. And that looks really simple, but we do this with thousands of larvae. We do it over a range of temperatures, and we do each temperature multiple times, so we have nice repetitive data. But what we can do is actually measure their thermal performance curve. The reason that matters, if you want to know, is that thermal performance curves are very, very good predictors of their ability to tolerate climate change. And we would like to know that. We also want to make sure that if we're going to be breeding across populations that are what we call crossed offspring, are going to have just as good thermal performance curves as some of these pure strains of species. Oh, the video is going to play again. There you go. All right. We can also use all this data, which we call ecophysiology. So eco meaning like the environment, how that impacts the physiology of the organism itself, to predict their extinction risk. So we can use all this information twofold. One, to know where to release to, like threefold actually. Two, to make sure that animals have the same fitness levels. But also three, we can use it to predict extinction in these species, and more specifically, even certain ponds. Oh, see, I'm ahead of myself again. This is the slide I was just talking about. So what we ended up doing is comparing these kind of pure pure populations. So the larvae that came from like a Santa Cruz pure strain to Monterey pure strain to the crosses, and see if they have the same level of survival traits, so that like growth, egg success, and also if they have the same level of thermal physiology. And if that's true, if they're all the same, then we can actually release the animals. We consider them equally fit, they are safe to release into the environment, and they're allowed to move on. So I'm going to go through some of our results. And the first is just about their natural history. I've said this many times already, but we knew really nothing about these adults except for how to find them before we brought them into the lab. We know a little bit more than that. We don't know very much else. There's a lot that's been assumed of the species based on its other related species, but not about the species in particular. So a lot of the data that we collected was just what do these animals do? So this is the female post her breeding. She's hanging out in her little tank with some dirt. We figured out that females lay approximately 100 eggs each. The female choice in this species is very, very strong. So to remind you, that is, females in this species have to get to, I guess, choose their mates. The mating system in the species is really fun, I think. But a male will approach a female. They actually lay their sperm in these little, what we call sperm packets on the ground. And then they do this little like wiggly dance to try and convince the females how awesome they are and to dance over their sperm packet. And then the females would pick up the sperm packet inside their cloacal and fertilize their eggs. But what that means is you have to be a pretty awesome male with a pretty awesome dance to convince the female to pick your sperm over somebody else's. So we played around with some ratios, like if you put one female in a breeding tank with one male, will they mate? The answer is no, not usually. A female needs at least 2 or 3 males to compare to pick the best one to even want to breed at all. We also found that the egg incubation period was about 3.5 weeks. So that's how long it takes from the eggs from being laid to when they hatch into larvae. And in our tanks, the larvae metamorphosed into adults in about 4 months. And unsurprisingly, we learned about their diet, as they pretty much want to eat anything. But they did have a preference for Daphnia, which is a small aquatic invertebrate. We also have, these are just some simple graphs. We have no difference in egg fertilization rate, egg mortality, or larval mortality, which is all great. We do have some differences in larval growth, which we deemed was due to density of larvae. So larvae kept in tanks with more larvae grow slower because there's less food per larva. Makes sense in retrospect, but when you're looking at your data and you don't understand, it confused us for a second. But at the end, when we kind of separate per density, larvae almost all grow about the same rate, which is great. So all larvae, no matter what of their parentage, have similar what we call survival traits. We also saw that there wasn't a lot of difference between larval's thermal preference. We see that almost all larvae have similar preference for temperature. They all prefer to be at the same temperature. That's great. It's also great that these temperatures all align with what the ponds were when we released the larvae into them. So we checked the temperature of the water before all these larvae were released, and they aligned well with the temperatures that they want to be at. So I was going to talk a little bit about the releases, because we actually were able to breed a lot more larvae than we thought we would be able to. So over the past 2 years, we've released almost 3,000 larvae into the wild, into all sorts of ponds. I should pull up that map again to show you exactly where. But we released them all into the struggling populations, so all into Monterey County. We released a bunch into the pond that we took adults from that was struggling. We released almost 700 into a historically used pond so that we know larvae and animals have been using but haven't been able to get to for a while. Fish and Wildlife made a whole new pond from scratch that we released a bunch of animals into after they tested the pond was all safe and good for release. They also reinforced some ponds. So some ponds that were natural, but with climate change have been draining too fast. They reinforced the bottom so they hold water longer. And we released a bunch into there. And then we also released some into a very small man-made pond where they're not found currently to see if we could reintroduce them. So this is really exciting. You can see these are all fish and wildlife biologists and me loading up larvae in buckets in their cars. The reason this is so exciting is not normally, but very frequently in conservation biology, you're not allowed to just move animals around willy-nilly. But the reason we're allowed to do this, besides that we've determined the species desperately needs it, is this was a huge collaboration between fish and wildlife. There are different fish and wildlife officers who run or are in charge of certain ponds, and they all have different biologists working under them. So it's kind of this collaborative event to do all this, keep track of it all, and release all of these larvae. You can see here at this pond, these are buckets that have a couple hundred larvae in them. They get set in the pond for a while to make sure they're the same temperature. Then a little water gets added to them, just like you would if you introduced like a fish to a new fish tank. And then the larvae get released. One of the cool things that I'm also going to talk about a little bit today is we also, before we released these animals, is we took a tissue sample from each of them. And I'm going to talk about why, but first I'm going to show you what that looks like. So these larvae, when they release, are about, 5 centimeters in total length. And what we do is take a little tissue sample from them for genetic analysis. And what we do, how we do that is kind of strange, but we take a little clip of their tails. So this species can regenerate their tail, especially when they go through metamorphosis. So they're larvae now, they'll go through metamorphosis to become an adult. And there's been many, many studies that show that this actually has no negative effect on the larvae being released or their survival rate. So what we did is we took a tail sample from all 3,000 larvae that were released. And this is just a video of how that works. And I'll talk over it as it plays. These are all samples, but a larva gets picked up. There we go. Into a net. It gets held, its tail gets clipped. Don't worry, it's very quick. It gets put on a piece of blotting paper to dry out. Larva gets put into bins for release. You can see these are much bigger than some of the photos I was taking earlier. And the tissue sample gets saved in a dried state. So these are larvae that are about to metamorphose any day now. And you can see our kind of operation. We have thousands of larvae going. And each of these little packets holds one tail clip and their ID label. Let's see. We're gonna watch it again. All right. Clip. It gets put on a piece of blotting paper. It's like, it looks big in this photo, but it's like 1/4 centimeter long. It's very tiny. All right, we'll stop the video there. Oh, it stopped itself. And what we can do now is run a genetic analysis. So, we don't want to watch the video again. I'll talk about the genetics in a second. Let's first talk about the results since the release. We are not 100% sure how adults know what pond to go to. We don't really know how adults know to come back to the pond that they were born in. We assume it has something to do with where they're born sets some sort of signal in their brain of how to get back to. So what we really wanted to do is make sure we release the larvae while they were still larvae, so they can acquaint themselves with that pond so they know to come back to it later. So another researcher doing a whole different project very kindly tracked the ponds that we were releasing in to see if any of our larvae actually successfully made it out of the pond and into adulthood. And they did, which was very exciting. They found hundreds of our larvae having successfully stayed alive in their pond until they went through more metamorphosis, successfully go through metamorphosis, which can be a dangerous thing, and make it onto land and start to migrate towards the forest. They were able to track a bunch of our larvae. And at the historic pond and the man-made pond, they counted, like, as I said, hundreds of our larvae leaving. And they weren't doing like full scale counts. That was just the fraction that they found. So that's really exciting. We know that our larvae are able to survive when they get dropped in the pond. They're also able to survive into metamorphosis and into getting onto land. So the kind of conclusions of our study here are that there was no outbreeding depression that we saw between the populations. There were no negative effects of breeding across the healthy the healthy Santa Cruz to the struggling Monterey populations. As I just said, larvae are able to survive being dropped into the wild from the lab conditions and reach metamorphosis. And then this kind of next step, and what I'm going to talk about now, is the genetic crossing that we're doing as a mechanism to save species that are facing extinction vortexes. So if I draw you back to the beginning, where I talk about extinction vortexes, the Santa Cruz long-toed population is very small. It might lose genetic diversity, which we already know it has, which means it's not capable of surviving disturbances and issues like climate change. And that cycles. So the only way to break the cycle at this point was to increase the genetic diversity. And so what we did is a real life test of, does genetic crossing and increasing genetic diversity break the extinction vortex? The answer to that is we'll have to see, and that's what I'm going to talk about next as our next steps. So we did repeat the breeding in 2022. We did not do it this year, but we will do it again next year. And what we're working on now is developing a protocol to analyze all of those tissue samples. And what we want to do is track the success of our animals. So we know that just adding a bunch of animals to the population will probably increase the size of the population, but we want to know if it increases the genetic diversity of the population. And we also want to know if our larvae, who are lab bred, can reach all the way to being full-grown adults and reproduce again. So what we're going to do is use all those tissue samples and develop what we call a heredity protocol, where we can track our baby larvae's future offspring. So they're going to create pretty much a family tree. So what we can do in a few years, when our larvae will come back to these ponds as adults, is catch all of the wild larvae, once again take some tissue samples, and see whose babies they are. Are the babies being born from the animals that were natural to that pond, or are they babies being born from animals that we released? And what that will let us do, besides knowing if this method works, and if it does increase genetic diversity, is we can literally track what we call the reproductive success of our larvae that we've translocated to different sites. And we can also track it by their cross type. So if we release pure Santa Cruz larvae, pure Monterey larvae, and cross larvae into one pond, we can see which three of those genetics are the strongest for both survival and reproduction. So we can alter our future breeding plans as well as test how what we did already worked. All right. And as I said, these are the kind of main three goals: is, are our animals successful? Can they breed, reach adulthood, and survive in the wild? Which genetic lines are more successful? That will help us in the future. Fish and Wildlife is planning to breed these animals pretty much forever at this point, is the plan. They're going to open up a full center soon, hopefully, to do this. And then 3, we want to know if we're increasing the genetic diversity. Are we breaking the extinction vortex? So these are kind of like the larger scale goals of just the breeding itself. But this is going to take a couple years for us to figure out. What we are still working on is crossing with some more populations. So we only cross between the first year 1 Santa Cruz population and 1 Monterey population, but we want to add in a bunch more Monterey populations. We are going to do something where we continue this kind of larval diet study and track, we call it trophic cascades. These larvae are actually some of the biggest predators in their little ponds. We don't have a lot of native fish here, so they are the top predator in their food chain when they're in these ponds. What that means is they can have great impacts on the lower levels of the food chain. So we're trying to design this experiment where we're using the diet data we collected to understand the cascading effect that these top level predators have on the other organisms in their ecosystems. We are actually tracking the population size. So Fish and Wildlife is going to keep some of these population studies growing and just see if we physically increase the number of adults that are coming back and forth to these ponds. And that is all to answer that question about, can we eliminate extinction vortex? We aren't always able to answer such questions since those are such a large scale question, but because the species is such a small range, we can monitor the whole thing and track genetics of the whole species, we can maybe actually answer this question. I'm hoping the answer is yes, and I'm excited to see how that goes. The other thing that I just wanted to point out here is endangered species have lots of protection around them. And what that means is most people are not allowed to handle them or touch them or see them or anything like that. There's lots of permits involved. However, this was a very big endeavor, and Fish and Wildlife allowed us to be really flexible and bring in more people than would normally be allowed. And what that means is we are allowed to do a lot more outreach when it comes to endangered species than we can normally do. And what that means is this picture on the right is some of the undergraduates at UCSC. A lot of them were able to come out and work in the ponds and get hands-on experience with endangered animals, which is very, very rare. Normally that's a no-go, but here they are able to help. It's very exciting for them, also very helpful for us. We're also able to have a lot of photographers come out. I know that sounds silly, but the species, first off, is hard to find, and second off, it's technically illegal to touch one. So most photographers and like natural history books that talk about the species don't have a picture or don't have a good picture. So a lot of photographers came out to get photos of these animals for their kind of their ID guides, their field guides. So we'll have some updated photos of these. We got a good collection of them. I showed some of them throughout the slide today. But it was a lot of fun. We got to have some high schools come and give tours. We were able to do a lot more outreach than we would normally be able to do. So that was really exciting. I won't go over this too much, but as you can imagine, there are so many people that have to help with this, not just on the UC Santa Cruz side, but also on the Fish and Wildlife and the reserve side. So I want to give a shout out to all those people. This is one of those photographers for pictures from one of those photographers up here. It's one of the adults right here. You can see how clear that is. You can even see like its eye and the shine on it. So we don't usually get pictures this great. And with that, I'm going to take questions. I tried to answer some as we went, but I didn't think I made them all. But thank you very much. And I'm very excited to kind of answer anything you want to know, either about this species or any of our local amphibians.
Regina, I'm going to monitor the chat in just a moment, but can you talk to us a little bit about parasites and if parasites, or what like recreationists who know that they're going to be down in the wetlands, like in their range, if there's anything that they can do in anticipation of being near their breeding ponds?
Yes, that's a great one. So it depends on where you are, but here in our county, we have two concerns of things we're trying to prevent from spreading. One is a parasite. It's a type of nematode. That's not dangerous in low levels to amphibians, but is deadly in high levels. So we're trying to prevent that from transferring. The other thing that we're trying to prevent from transferring is chytrid. A lot of people may have heard of chytrid, but it's an amphibian bacteria. Yeah, I keep forgetting if it's a fungus or bacteria. But it's a disease. It's very deadly for all amphibians. Some amphibians can fungus. Thanks, Brandon. Blowing my mind here. I always confuses me because its name is like bacterium something or other. It's a fungus. Thank you. Chytrid fungus is very contagious and very dangerous. And once it's established in a population, it's kind of stuck there. So those are the two things we're trying to prevent from spreading. So the big thing that you can do is to, the summary is we don't want to be in one water source and then bring the same equipment or shoes that we're wearing to another water source. So if you're going out just for the day and you're going to just one area, you're good. But if you're going between a bunch of areas, then there are proper disinfecting procedures that are supposed to be followed. I won't go through exactly what they are, but I could send Dylan that if people want to know. But yeah, the big thing we're doing is that, to be honest, you're probably not going to be able to get to long-toed salamander ponds. Every single one that I've been to has at least one gate in front of it, if not a couple gates, or is on private property. So you will not be able to actually go to the breeding ponds themselves unless you have a no with somebody on the inside. Does that answer your question?
Yeah, absolutely. Thank you so much. And that also just seems like a good habit to follow if you're traveling between water bodies. And so I would encourage everyone to heed Regina's advice and share that with others. And I'm going to ask one more question. I apologize for everyone for somewhat monopolizing the conversation here. Regina, if you could somehow gather everyone into the same room and be able to tell them like one sort of big picture idea, like what would you want those folks to leave the room knowing and being eager to like share with other members of the public and such?
Ooh. Well, when we talk specifically about this species, I think you all probably know because you're nature enthusiasts. But a lot of people don't know that we have our own local endangered salamander here. So bringing alertness to it and its plight is really helpful. I make it sound like this big collaborative event, which it is. But there's also a lot of opposition on projects like this. There's a lot of private landowners and agricultural people who don't really want to do what's right for the salamander because they have other interests. So there's a lot of work with just community engagement. So one of the best ways that we can do is just make people aware of what's happening, that we have our own special salamander that's just found here and that we want to protect it. And bringing just awareness to that would always be helpful.